this is going to be quick. We're going to have 5-minute max brief presentations by each group on like how they would advance the specific focus area that they picked. And then we're going to have a final vote. And let's see who gets the master prize out of all of them. Okay, first one. Who here wants to present on live cell age clock?
We propose genetically encoded age indicators, so they should be in vivo, also single cell resolution, fluorescent, and multiplexed, so that means that we can take into account like many different markers simultaneously. So they would be based on CpG methylation sites and also other biomarkers of aging. And the most important idea for me here is that they would be monitored online, and that means that we not just monitor them, but we also potentially can control and probably reverse them online. And yeah.
Awesome, yeah, thanks. So we had several different challenges to solve, right? And one kind of the biggest challenge would be to do this in vivo. We also thought of some intermediate steps to go with organoids first, and we had several different ideas to tackle this challenge in organoids. So to not have to harvest the organoids, but do time-based organoid measurements on a single cell basis. One of the ideas was actually Mark's, but he didn't want to come up here, so here I am. It's an RNA-guided methylation site detector. So basically, if a methyl is present on an mRNA, the mRNA is released and it produces a fluorescent protein, which is a great readout, obviously, in any high-throughput setup. And then the second idea we had was exosome-based readouts, which obviously allows sequencing if you have DNA in there. The difficulty is to get the DNA in the exosome, right? So there are some systems that allow to bind proteins to exosomes and form them that way. And DNA binding proteins have also been engineered that would capture, for instance, plasmid detectors in the cell. You then have exosomes being formed and outputted, and you can sequence the content of those exosomes, which would allow for a readout without destroying the cell. And then finally, we have a great idea that's highlighted by Anna.
There was a paper published in 2024 showing that you can record events in a cell on a DNA ticker tape using prime-edited guide RNAs. So one, particularly as an adjunct to the fluorescence idea, is you can have methylation of certain regions, trigger editing of a piece of DNA where you that encodes surface proteins, and so methylation of a given site will insert a promoter at upstream of one of these genes that is encoding a surface protein. So you can have surface protein expression contingent on methylation of particular sites. And then there is another paper that I read a while ago showing, demonstrating that you can use nanoparticles to target surface protein expression that allows you to do in vivo monitoring of those protein expressions if you tie those nanoparticles to fluorescent reporter or something along those lines.
So to wrap it up, lots of work to do. Please vote for us.
I want to comment also that because like I proposed and Yelena proposed, this was a kind of joint paper there on this idea, but what is important that we need multiple signals, not just one signal, because like in aging clocks we have 100 or more CpG sites or many genes, for example, in transcriptomic clock. Here, I think we could maybe put in a cell up to 3 signals, maybe based on fluorescent proteins that would cover a certain wavelength, up to 3. But 3 is not enough, maybe, but that's what can be done. So my point is that more than one signal is important, not just one signal.
Actually, it could be put even more than 3. We can multiplex that, and it's done in special transcriptomics, for example. We can make a lot of sites.
Oh, thank you. Okay, next one is high throughput cryoprotectant screening. Okay, take it away.
So I'll stand over here as well. Thanks. So the idea we workshop was developing a high throughput or automated discovery workflow for better cryoprotectants. So to enable practical, cost-effective biostasis, you need a cryoprotectant that gives you as low as possible a critical cooling and warming rate, while also not being toxic. Current best available chemicals, toxicity is okay, but the critical warming rate is, on the order of 10 degrees per minute, which is incredibly difficult to achieve on something as large as a human body. So the idea here, is to develop these automation tools in the fields of organometallic catalysis. There are already unit operations that have been developed for cooling and warming of chemicals down to the temperatures that are needed for cryoprotection. You need this for all sorts of chemical reactions as well. There's also various assays that can assess crystallization, measure TG, freezing point, et cetera. So the idea would be to basically build out one of these automated workflows capable of screening on the order of 100 to 1,000 compounds per day, and then develop the analytical methods that you need specifically for cryopreservation, in particular the assessment of diffusion into tissue, tissue damage, as well as cooling and rewarming of small organisms like C. elegans. A workflow like this typically would cost anywhere between $5 and $15 million and about 1 to 2 years to develop, at which point you could then conduct a screening campaign to go through orders of magnitude more compounds that have been looked at today. And sorry, not just compounds, but actually full formulations, moving past the simple 2-component systems that are used right now. Lots of work to investigate the details of exactly what's needed on a chemical basis. There's tons of interesting dialogue between the people who actually understood chemical high throughput screening and the people who understood cryopreservation. A little bit more of that, even a few days, could probably get us quite a lot farther.
Wonderful, thank you. We are also wildly under time. Questions, comments, feedback, criticisms, helpful suggestions?
I don't know if this came up, but is there any way to maybe use extreme organisms that hibernate or that lower their body temperature safely as a way to, instead of having to use a chemical, do some kind of other intervention that naturally makes them more susceptible to the cryopreservation?
Yeah, so this is not something we discussed. Our focus was on accelerating the discovery of those chemicals. Personally, that sounds really interesting to me, but maybe, but as a chemist, that's also something I know nothing about.
There's time for one more if you want. Okay, wonderful. Thank you so much. Mark, do you want to announce the next two? Okay, I'll go ahead then. Next one up, we have embryonic rejuvenation to generate younger organs.
So this is the topic on organ replacement. And the idea is kind of convergence of two important developments in the field. So one is what was presented by Omri on this generation of embryos from naive iPS cells. Naive cells, they are differentiated into 3 germ layers, and then eventually they assemble the embryo at a day point 8.5, and then continue kind of development. And the second idea is what we call embryonic rejuvenation. And this is something that we found in my lab recently, where apparently there is a step of, it's the only natural rejuvenation process, by the way, happens in mid-embryogenesis, starting at the blastocyst stage, and then post-implantation, embryo achieves the lowest biological age, and that's where the aging actually begins. When we quantify aging based on any of the clocks, aging begins at that point. So I think it's important to kind of combine these two advances and to achieve organ replacement with the best possible organs at the youngest biological age. And for that, we need to study this very extensively. And so, for example, some ideas that we brainstormed is that maybe immediately apply like aging clocks to this experiment that was developed by Jacob Hanna on like assembled embryos, because we don't know if they achieve embryonic rejuvenation, if that step is bypassed, and therefore the embryos that I developed, they already a little bit aged, and therefore their organs would be a little bit older. So this is kind of the main idea. So study embryonic rejuvenation, try to understand mechanism, and apply this to organ to obtain organs which would be best for replacement. Yeah.
Awesome. Thank you. Question, comment?
So there is a very small period of time between the zygote formation and the implantation, and it's like 3 weeks. How much of rejuvenation happens during that time? Does it really matter if you have to maturate the tissue, grow an entire body in the acephalic replacement, for example? Is the organ going to be more youthful? Is it going to be more resilient if it goes through that reset point?
This is unclear, but naturally we all mammals seem to go through that transition. Rejuvenation transition, and we observe this in mice, in humans, and frogs. How exactly this happens is still not fully clear, although we have like many advances. For example, we know that all cells are rejuvenated except for the extraembryonic cells. We know like in terms of pathways and so on. But exactly, like mechanistically, the idea would be to transfer that information, and ideally would be to transfer also to adult organs, right? To partially rejuvenate them through this process. But mechanistically, it's unclear yet.
Do you think that whatever is causing that is different from Yamanaka factors, or somehow related?
Yeah, this is also actually one question that we discussed in our group. This is also unclear because Yamanaka factors, when they are expressed in adult cells, support rejuvenation, but also they are expressed in the blastocyst just before the rejuvenation step. It's possible that these factors, plus maybe some other factors, they also contribute to this process. But this is currently unclear whether they are essential for it. Because rejuvenation can also be achieved by other factors, like there is a completely different set of factors can be used to rejuvenate, or for example also chemical. One can reprogram cells through chemical approaches.
Thank you so much, Vadim. Wonderful. Last but not least, AI-driven surgical robots for replacement.
Business guy. There we go. One of the rare business guys here. I try my best. So yeah, we actually had the focus of an AI surgeon for replacement surgeries. And overall, we were just talking about, maybe to give you a bit of context about research. So because we all are not specialists in that field, so we have talked about Neuralink, actually how they do and why they not have automated it already. And so we came up that we talking about the doctors, right? And that normally doctors don't like normally being shadowed or also be recorded during surgeries. Long story short, we came up with a kind of VR solution to actually first go into testing and shadowing these kind of things, and to mitigate the risk actually of certain challenges from a legal perspective. And why we're going to do this overall is actually that we said, okay, it has to be much more scalable, because currently surgeons, it's already the norm that they don't have enough capacity in terms of we're gonna overall increase and scale these replacements, that they would have the capacity. It's not today. The second thing is precision, right? We need to have that in a certain ways. That's why I think also currently already Neuralink is doing it. And the last thing what comes with that is a really economically driven thing, is the price. So we first said that the solution is going to be most probably a supervised solution, yeah, and that we overall really have to also look into what was there. a bit of actually also ethical, their regulatory and ethical compliance. So where to start at is also quite still open. Something I missed, guys? Forgot to say? Research? What?
It would be a team that kind of wants to take steps in that direction, and we're happy to.
Oh, yeah.
Want to grant them
Yeah.
money rather than.
Yeah, correct. We were thinking about it, since we are not experts, to, in case something magically happened, we're going to win the prize, that we would love to grant that, actually make a bit of noise, right? Marketing side, maybe through the LBF, get a lot of actually people engaged in this topic and really get something done here, so that people are really working on this instead of it's maybe a lost opportunity.
Awesome. Thank you. Any questions? We do have questions.
All right. So if the AI commits malpractice, who's who's at fault here?
Yeah. Fair point. That's why. You're at fault. Yeah, it's always the business guy. No, actually, that's why we started to say it's definitely a supervised solution first. I think we believe that it's ethically reasoned that it will stay very, very long in that sense, until it's very, very, very clear and very, very precise. So, yeah.
Next you. Yeah.
So one suggestion to get started on this could be to create a simulation in which you are training a agent that is doing the surgery. And once you figure out, okay, it's working very well, then you could probably try to do some sim to real.
Yeah.
That could be one way to go about it.
Max, hello. Do you hear
And
me?
this is how most of the reinforcement learning is also done, so probably that makes sense to do.
You can probably also just project.
That's okay. Yeah, makes totally sense. But again, we had at least it seems like that we had an expert in that field. So there were a lot of actually field studies already happen, and really doctors don't like to be actually shadowed for also, I think, really reliable reasons. So I think that's why we came up with the VR kind of solution. But there's still even a mismatch, right? I think it's still actually, how do you really break down the workflow to create an AI agent in second space? So that's something I think that really needs to be researched first.
All right. Well, thank you so much. Wonderful. Wow, I have 2 mics. Dangerous. Okay. Okay. We're almost at drinks, you guys. You've almost made it. Last but not least, Mark has taken the time to actually write so basically those are the top 4 projects again, and he just wrote them up on a new sheet next to it, especially because some of them changed a little bit. And so you have 1 more vote to do tonight, and the sooner you do it, the sooner you have drinks and food. So you have 1 black dot this time, so cast it for 1 project, ideally not yours, and run up here. Let's go.
All right, we're going to announce the winner, guys. Are you ready?
Hey everyone, are you ready?
Are you ready? So, I feel like maybe we need to redesign this in the future, because somehow I think Vadim's on all of them, so he wins no matter what. But Vadim won. No. Embryonic rejuvenation to generate younger organs is the winner. Yeah. And the prize is going to be $1,500. We recommend using that to perhaps allow somebody to spend the time to write up a more detailed proposal that we could float to funders that we know. So that is that, and I guess the winners, who was in support of this? Whose team was it? Come up here so we can give it an applause. Come on up. All right, everyone, a round of applause. We got through a very long day, a very busy day. You guys all worked really hard, and I think the finalists were all pretty cool. And we have another date, so we'll be seeing you tomorrow.